Thank you for joining us today for the Kirby Institute seminar series. My name is Miles Davenport. I'd like to begin by acknowledging the traditional custodians of the lands on which we gather today and pay my respects to their elders past, present and emerging. In particular, I'm speaking today from the lands of the Cadigal people of the Eora Nation. I extend that respect to Aboriginal and Torres Strait Islander peoples joining us today. Some housekeeping to start off with. The format of the seminar will start off with a presentation, followed by a Q&A at the end. To ask a question, click on the icon with the speech bubble and question mark in the top right corner to open the Q&A chat panel. When you click on Ask a Question, please remember to write your name. This will help us reference your question and make answering easier. So today I'm very pleased to welcome Professor Katharine Bar from the University of Pennsylvania. Katharine is a physician scientist and clinical trialist who studies transmission, pathogenesis and persistence of HIV-1 and other pathogens. She's been involved in a number of trials of antibody therapy and also understanding immune escape from antibody recognition. And more recently, this work has extended to obviously COVID, as with many other people. But today, Katharine's going to talk to us about lessons learned from HIV-1 rebound after antiretroviral treatment interruption. So I'm sure you'll join me in welcoming Katharine.
All right. Thank you so much, Miles. I'm joining you from later in the evening in Philadelphia in the United States, and it's just a real pleasure to be invited. So thank you very much for the opportunity. So what I wanted to talk about today are some of the studies that we've done characterizing viral rebound in the context of treatment interruption. And so as many of you are aware, if we think about a person living with HIV who has viremia and then they start on antiretrovirals, when they start on antiretrovirals, we see a classic sort of decay of their virus until it's underneath the limit of detection of our clinical assays, and we sort of have this chronic steady state of the latent reservoir until treatment interruption. And treatment interruption happens oftentimes in people's own lives for whatever reasons, but we've been doing it more recently in the context of research and treatment interruption studies, and that's what I'm going to talk about. So we know when we interrupt therapy, virus does rebound, and this viral rebound and the virions that are replicating systemically that we can sample are what I'm going to talk about again. So the reason we think rebound is important is because it is a direct reflection of the clinically relevant latent reservoir. So this is the reservoir that is able to replicate and actually restart infection after treatment interruption or another context. It is also, viral rebound is also the clinical juncture at which suppressive or curative strategies will be either implemented or tested. So it's important to understand all the virus and host dynamics that are at play in this scenario so that we can improve the chances for cure strategies or suppressive strategies to be effective. So treatment interruption has been conducted for almost as long as we've had combination antiretroviral therapy. So this is actually a publication from 1999 from an early treatment interruption study. And what you can see, each one of these lines represents the viral kinetics of an individual who interrupted therapy. And what you can see from all of these, from this picture, is that as soon as treatment interruption occurred, within about 1 to 2 weeks, all of these individuals had virus return, and that is been consistent in studies since this time more than 20 years ago. So if we look at meta-analyses of many treatment interruption studies, what we see is that rebound after treatment interruption is almost universal. Within 2 to 4 weeks, we see more than 85% of individuals have virus return. And then by 8 weeks, more than 95% of individuals have virus return. There are, of course, a few individuals who maintain suppression, post-treatment controllers, and there's a lot of research into those groups, but we're going to focus on them a little less today. So one of the few things that we do know about rebound is when this occurs, we know that multiple virus populations return, such that virus is polyclonal, and we see multiple genetically distinct lineages. And we also know that suppressive or eradicative strategies can delay the time to rebound and or reduce the number of these lineages. However, the origin of rebound virus and the biology of rebound viruses, as well as the host immune pressures that govern this emergence, are still somewhat unclear. And these are some of the questions that myself and my colleagues have been studying over the past few years. So one of the first studies that we were able to really dig into this with was an ACTG, or an AIDS clinical trials group, and this is the USA's clinical trials network for people living with HIV. So a study called A5340, and this was a study to ask whether VRC01, or one of the first clinically available broadly neutralizing antibodies, when given in the context of a treatment interruption, could maintain virus suppression. And so the schema was pretty straightforward. We had 14 participants in this study. They stopped their antiretrovirals. They were given 3 doses of the antibody, and they were followed for a total time period of about 8 weeks to see whether we could maintain suppression. And so here is the viral load curves from these individuals, and it looks a lot like that picture I just showed you of a treatment interruption and viral rebound without any intervention. So unfortunately, there wasn't a major change seen here, in that most of the individuals experienced viral rebound relatively early, within sort of 2 to 4 weeks. And so indeed, all participants experienced viral rebound between 2 and 11 weeks after treatment interruption. And if you factor in these 2 more delayed rebound experiences, we did see a modest, statistically significant delay in rebound versus historical controls, but not really much of a clinically significant durable suppression. And the reason we determined that this antibody was unable to maintain with suppression was because there was prevalent pre-existing resistance to VRC01. And so I'm just going to briefly show you some of the studies we did to figure this out. But here, if we look at individuals who rebounded quite early, and we sequence the viruses that rebounded here, and we compare them to viruses that were circulating pre-initiation of antiretrovirals years earlier. So in black, we have the pre-ART viruses, and in red, we have rebound viruses from week 1, and in orange, we have rebound viruses from week 2. What we can see is that compared to the sort of diverse virus population represented by the black sequences of chronic viremia, we have multiple rebound variants, multiple genetically linked populations of viruses that are rebounding to cause recrudescent virus at treatment interruption. And if we clone these envelopes and we test them phenotypically to see what their sensitivities are to the antibody that was tested, VRC01, what we see is that we have evidence of resistance within the baseline virus populations, and that the viruses that emerged were also quite resistant. So across the board in these early rebounders, what we saw was the existence of baseline resistance, as well as rebound viruses that were quite resistant to this antibody. Now, in contrast, if we look at our 2 individuals who had delayed rebound, if we look at the viruses that were baseline, or the sort of pre-existing pre-ART viruses, what we see actually is much lower IC50s, or a greater sensitivity to this antibody. And indeed, the single or 2 lineages of viruses that rebounded at the time of treatment interruption had, again, more sensitive virus that emerged after the antibodies had waned from the system. So this antibody worked when the reservoir was quite sensitive, and it did not work when the reservoir was resistant. So the conclusion clinically was that VRC01 monotherapy was unable to durably suppress rebound viremia in the majority of participants due to the prevalence of pre-existent VRC01 resistance. We also saw that the treatment interruption was clinically well tolerated, but one of the things we wanted to ask in a little more depth was, did it alter the latent reservoir? And so we went on to do a second study based on this clinical trial. So after the initial study here, we followed these participants for an additional 6 to 12 months, about 8 months on average. And in addition to looking at the pre-ART plasma virus and the rebound plasma virus, we went ahead and looked at leukapheresis, or PBMCs, cells collected from the blood prior to this trial, and then 6 to 8 months after virus had been resuppressed on antiretrovirals. And so at this point, we did, our goal was to see whether the reservoir was altered by this study. Now, when we initially planned the study, we were optimistic that this antibody maybe would have wonderful effects, and through antibody-dependent cellular cytotoxicity, perhaps it would effectively reduce the size of the reservoir. But given the fact that there is so much resistance and very little effect of the antibody in maintaining suppression, we now wanted to ask whether the treatment interruption in and of itself actually increased the size of the reservoir. So we wanted to make sure we had not done any harm with this treatment interruption study. So we wanted to do both quantitative as well as qualitative comparisons of the reservoir before and after the study to see if we had changed the composition of the reservoir. So the first thing we did were quantitative measures. So comparing pre-ATI and post-ATI CD4 positive T cells, we quantitatively assessed the total proviral DNA. And if you look from before to after, you can see some of the lines seem to go up, some seem to go down, but in general, there's very little change in the size of the proviral DNA reservoir. Similarly, when we looked at cell-associated RNA or transcriptionally active virus, we see no substantial differences in the size of the transcriptionally active reservoir. And finally, we performed quantitative virus outgrowth assays, or QVOA, to assess for replication competent virus either before or after. And again, we do see some participants going up and some going down, but in general, there's no consistent or substantive change in the size of the reservoir by any of these measures. The next thing we wanted to ask was, if we look at the sequences that make up these reservoirs, are there phylogenetic differences? Are there sequence differences in the quality of the reservoir? So what I'm showing here is a phylogenetic tree from one of the participants. And again, we have our pre-ART virus in black and our rebound viruses in red. But what we've added is looking at those QVOA, those quantitative virus outgrowth assays. If we sequence the virus from each one of those wells and we add them into the tree, we can say, what do the sequences of the virus in the replication competent reservoir look like before and after this trial? And one of the striking things that we found, which is similar to many other reservoir studies, is that a large chunk of the reservoir was indeed a part of identical virus sequences. And the most likely, the most plausible explanation is that these are expanded clones that's well characterized to be a major component of many people's reservoir. So in this particular individual, A09, who had quite a diverse virus population, the reservoir had about 45% of its sequences that were sampled more than once, so part of an expanded clonal population. And if we compare the blue pre and post-ATI QVOA sequences with rebound, multiple rebound lineages, what we can see is there is no overlap between these two populations. So although they're similar and they interdigitate, we have not sampled the same viruses in the reservoir that would ultimately go on to rebound. So here's another example of a different participant. This participant has a smaller, a less diverse reservoir, but when we look at their reservoir sequences, again, we find examples of expanded clones here. And then if we look at our single rebound population in this individual, again, it is distinct from either the pre-ATI, but most importantly, the reservoir sequences here. And so what we saw across the board with our participants was that the reservoir, both before and after the trial, and the rebound viruses did not overlap. They were not duplicating each other at all. Now, there's always issues with sampling when it comes to sequencing. And in particular, we are doing a relatively limited number of sequences for a heterogeneous population. So the reservoir is large and diverse, and so we're clearly undersampling the reservoir when we do these types of QVOA assays. But there's a few participants in which the reservoir sequencing was so homogeneous that it gave us more statistical power to be able to say we were capturing or characterizing in general what the reservoir looked like. So this participant A01 is fairly interesting because it has a hugely expanded clone that takes up the vast majority of the sampled QVOA or replication competent reservoir. So we did about I think there's about 50 sequences here, and I'm not going to count them right now, but there's somewhere between 40 and 50 sequences. And again, these filled boxes are before the study, and the open boxes are after the trial. And you can see they are genetically identical to each other. And so this represents one huge expanded clone that really embody the vast majority, 95% of the sampled reservoir. And yet, when we look at the rebound viruses, we have 4 distinct virus populations that replicated at treatment interruption, and none of these is represented by this huge homogeneous clone that's part of the reservoir. And so from some of these populations with large expanded clones, we were able to have more statistical power to say that we had not that we had sampled enough to show there were true differences between the reservoir and the rebound viruses. So to further sort of deal with sampling issues, we went ahead and used another method for sampling the reservoir. So looking at the same cells that are the input for the virus outgrowth assay, so resting CD4 positive T cells taken from the blood, we directly sequenced the envelope genes of those viruses from the proviral DNA of these cells. And so this is kind of a messy phylogenetic tree, but it's the same patient I showed you before with this one large expanded clone of the reservoir. But these purple sequences from before and after have now filled in a lot of sort of diverse virus populations within this phylogenetic tree. So they're clearly related, but there's a lot more diversity. It may be hard to pick up here, but there's quite a few identical sequences within the ONV proviral DNA as well. So we have many expanded clones within the intact envelope sequences. But again, if we compare them to the rebound viruses, there is zero overlap. We do not find any identical ONV sequences in the rebound or the proviral DNA intact ONV sequences. And if we do our little donut plots to sort of show what these virus populations look like, with our QVOA, we have in red this one large expanded clone, as well as 2 other populations that are sampled. With our ONV intact viruses, we see that red clone is a minor but sampled population. This blue clone is a little bit more prominent in the intact ONV, but they're quite distinct from each other. And none of these represent the 4 rebound virus populations in this individual. So we did one more step of sequencing in that we took those same resting CD4 positive T cells and we spent a lot of energy trying to sample just intact whole genome sequencing. So this is near full length sequencing. It's a little over 9 kb, so 9,000 nucleotides, where all of the genes are intact and the important elements are considered. And what we can see from these sort of 70 near full length intact sequences was they interdigitate within here. We can see that there's about 20 of them that fall within this large expanded clone. Another large chunk of them fall within this expanded clone. And so we again we see clones of these intact proviral DNA sequences. And if we look at our DNA plot or donut plots again, we see there is some similarity between our replication competent virus and our intact full genome viruses, but they're also slightly different. So our sampled reservoir differs based on the method that we use. And these are all reasonable methods that are used by many investigators when they're characterizing the reservoir. And yet in this particular individual, I think you can really see how you can have quite a diverse representation of the sampled reservoir. But importantly, regardless of which method we use, we are not accurately identifying the viruses that then go on in this individual to reactivate during rebound So I just want to mention that several other groups around the same time as we were doing these studies did very similar studies in another group of clinical trial participants who underwent monotherapy with a CD4 binding site targeting bNAb. So this is work from the Nussensweig group and clinical trials led by Marina Casti from the Rockefeller in New York, and they looked at 3BNC117, which is a very similar antibody to VRCL1. It's in fact comes from the same HIV positive individual. It's from the same clonal lineage. It's just a bit more potent than VRCL1. So they did similar studies looking at whether this one antibody could prevent treatment or could prevent rebound in participants undergoing a treatment interruption. And they did very similar methods to us in terms of sequencing the reservoir before and after the treatment interruption and comparing it to rebound. And when they looked at individuals with either antibody-resistant or antibody-sensitive virus, they again did not see overlap between these two virus populations. The Rockefeller group also recently in the last year did a study where they looked in a treatment interruption trial, and instead of just looking at the reservoir that circulates in the blood, they also looked at some reservoir cells from the lymph nodes. And they showed there was very tight overlap between the reservoir that was sampled from the blood and from the lymph nodes, but it was really quite distinct from what they found in the individuals when they underwent a treatment interruption and had rebound. So again, both groups kind of recapitulated the same themes. The nuisance white group has also implicated recombination as a potential mechanism for the distinction between rebound and the reservoir viruses. But other than the recombination issue, the findings are very similar from the BRCL1 and the 3BNC studies. So what are the implications of the distinct sampling of reservoir versus rebound viruses? Well, from a clinical trials perspective, which is of course something I think about as we're attempting to design new antibody-based clinical trials, it first of all means that our pre-screening methods that we have available to us are just not predictive. So they may be helpful in understanding the general constructs of someone's reservoir, but we are not able to predict the specific viruses that are likely to reactivate and cause systemic replication upon treatment interruption by sampling the reservoir through either virus outgrowth assays, or sequencing of provirus. The other important clinical consequence, I think, is when we compare the before the trial and after the trial reservoirs, and they look very, very similar to each other. This gives us some confidence that there was no substantial reseeding of the reservoir during this very short ATI. And I should mention that there was one participant in our study who we did sample some, or we did see some reseeding of the reservoir, and I'm going to come back to that later. But with the relatively limited sampling of the post-ATI reservoir, we did not see massive changes either in the size or in the quality of the reservoir. So that gives us some comfort. But the distinction between the sampled reservoir and the rebound viruses also suggests that rebound viruses come from someplace other than the majority of the circulating reservoir that we are sampling. So perhaps this is a compartment, the central nervous system, or other compartmentalized areas. Perhaps this is in a non-CD4 T cell type cell. Maybe it's in macrophages. Maybe it's in some other non-circulating or a tissue-restricted cell. Anyway, it leads to those sorts of possibilities. Another possibility is that perhaps at the time of treatment interruption, there are intense immune pressures that are selecting for only a subset of viruses to be able to reactivate. And these selective pressures are either driving adaptation of those viruses from what was present in the reservoir, or they're selecting rare variants that are uniquely able to reactivate. And so for the next part of the talk, I'm going to talk about some of the immune dynamics of rebound that we have been looking at. So the first question we asked is, what is the role of autologous neutralizing antibodies? Now, autologous neutralizing antibodies are very well characterized in other clinical junctures. So we know that autologous neutralizing antibodies arise within a few weeks to months of infection. They are not capable of clearing infection in the vast majority of individuals, but they drive selection of virus populations. The virus is always one step ahead of the antibody response in general, so the virus quickly escapes from the nascent antibody response when there's this back and forth of waves of antibody responses and waves of virus escape. And this is very well characterized throughout chronic infection. As long as there's ongoing viremia and there's continued antigen driving those antibody responses and the virus replicates, it is able to quickly escape. However, when you start antiretroviral therapy and you suppress all of that antigen and you theoretically remove some of the exhaustion of the immune system by getting rid of that chronic inflammation, we don't really know exactly what happens to those antibody responses. There have been some studies that show that they wane over time with antiretrovirals, which makes sense, but we don't know to what extent they wane universally, and we don't know which lineages or which viruses those antibody responses remain potent against. And so that's one of the things we were interested in studying. So in this A5340 study, one of the opportunities we had was to look at those rebound and reservoir and pre-art viruses and ask about their sensitivities to both the antibody we used in this study, VRC01, as well as the antibodies that were circulating, autologous antibodies. So here we're looking at plasma viruses in black, reservoir viruses in blue, and rebound viruses in red. And these are pseudotyped envelopes sampled directly from 4 different participants and then tested for sensitivity, in this case against VRC01. And what we saw was there was very little difference in the sensitivity of these different viruses to VRC01, suggesting that VRC01 did not sieve out a large portion of reservoir viruses that were inherently more sensitive than those that rebounded. In fact, there was enough resistance to VRC01 that it seems not to have changed the composition between rebound and reservoir. On the other hand, when we looked at the autologous plasma, so this is the plasma at the time of treatment interruption, we can see there's actually, in 3 of the 4 patients where we are sampling, there's a large difference. Now, this is just a statistical trend. It doesn't quite meet the cutoff for a statistically significant effect by most measures, but in all 3 of these patients, you can see that rebound viruses were in fact more resistant to the autologous antibodies than the reservoir viruses that were sampled in contrast from the same participant. And this suggests that the autologous antibodies present at the time of reactivation may have restricted some viruses and enabled others to be more fit in that context. So that's just one hint of energy, or one hint of evidence that autologous antibodies may have some role in this context. So there was also a study published by the Silicano group recently, and the Silicanos they're a lab at Johns Hopkins, and they collaborated with us on the A5340 study, and we collaborated with them on this study, asking if we use this QVOA system to test the effect of circulating antibodies, can we see that autologous antibodies have any ability to restrict or to block the viruses that could otherwise arise? And so what they showed when they took participants in whom they had serum and they took out IgG to represent the antibody pressure, and they tested whether the autologous IgG could prevent reactivation in the QVOA and the virus outgrowth format. And so what we're comparing is autologous antibodies, or the neutralizing antibody response, compared to no antibodies or a control IgG. And what you can see in these 3 individuals is that the autologous antibodies seem to block a great proportion of the viruses that were otherwise able to reactivate. Now, these are the 3 most sort of significant examples of the participants that they studied, and across their whole cohort, in fact, you can see a range in the percent of the viruses that were restricted by antibodies. But the point is there's variable but real ability of these autologous antibodies to suppress virus in this sort of in vitro or ex vivo context. So another question we had about autologous antibodies is whether they continued to have impact over the course of a treatment interruption or rebound viremia. So this is a historical, a relatively older treatment interruption study. It was actually conducted back in the early 2000s, when our sort of concept of what monitoring and duration of treatment interruption was a little bit different. So this was a study conducted by the Wistar Institute, which is a research institute also in Philadelphia. And they had 11 participants in whom virus was, or treatment was interrupted, and they followed viral rebound for a variable amount of time, but many, many weeks in these individuals. And so what we did was we took the first detectable plasma, this is usually between 2 and 4 weeks, the first detectable plasma viremia, this is between 2 and 4 weeks, and we sequenced the envelope genes. Here's 4 individuals. And like we've shown in these other participants, we see multiple distinct virus populations replicating at the first time of treatment interruption first detectable viremia. So we have, again, this polyclonal rebound that occurs. But then we ask, what happens to these virus populations over time? So here's that same participant. The tree is a little bit more spaced out now. We have 4 distinct, closely related, genetically linked populations. This is at week 4, the first sampling. And if we look a few weeks later with the same, the week 4 viruses shown here in black, and week 10, so 6 weeks later, viruses shown in green, what we can see is that some of these virus populations continue to be sampled. So here is this lineage right here where we have week 4 and week 10 viruses that are very closely related. And down here we have the same thing, week 4 and week 10 viruses continuing to be sampled over time. However, there are some, so these sort of lineages continue to replicate over weeks and weeks of treatment interruption. They persist. And there are other populations that were actually a sizable fraction of the first, detectable viremia, but they're no longer sampled later on. And so they've either been reduced or cleared from the virus population. And so if we looked across multiple participants, what we found, in fact, is that many individuals had early viruses that were circulating at a high frequency at first detectable viremia and were subsequently cleared. And we asked if there was an association between these cleared viruses and antibody responses. And so what we did was we took the envelope genes from these particular viruses, we made pseudoviruses, and we tested for sensitivity to autologous viruses circulating at this first time point and slightly later. And what we found in this particular individual is at week 4, when these early virus populations are first sampled, we see that there's about the same titers against these populations that ultimately persist or clear. However, once these, the action has happened and the viruses either persist or clear, we see that the viruses that were cleared have a substantial increase in the antibody pressure against them. So there's an association between increasing antibody titers against the viruses that are cleared from the system. And if we do this across these 3 different participants that I just showed here, we do see a statistically significant about 1 log difference. At the time that viruses are cleared, the antibody pressure against them is about the titers or IC50 reciprocal dilutions are about 10-fold greater against cleared viruses versus those that persist. So the next thing we wanted to know is, does that differential in antibody response towards the viruses that will arise and go on to persist versus arise and get cleared, does that predate the treatment interruption? And so we looked at this time point pre-ATI. So this is while antiretrovirals are still on board. We pulled out the IgG and we asked, is that IgG differentially pressuring the cleared viruses here versus the persisting viruses here? And what we see is actually about a 2 log or almost 100-fold difference in sort of the average responses against the viruses that later on would have increases in those responses and be cleared. And if we think about the sort of quantity of this antibody response, if we think about sort of classic amounts of IgG in the blood, this amount of a response is somewhere between 1 and 10 times the IC50 needed to clear these viruses that are circulating at the time of treatment interruption. So it's possible that this has an effect in sort of determining how these viruses either persist or not. Subsequently, we see that as the viruses shift, we see the pressures against them also shift and reflect sort of, increase in the cleared viruses, and then ultimately a shift as those viruses are no longer there. We see the responses towards the persisting viruses increase a little bit. So in terms of the autologous neutralizing antibody response, we think that the antibody response, there's several lines of evidence suggesting that it may modestly or mildly restrict systemic reactivation in some viruses. So in some participants and against some viruses of these participants. So it is not this universally blunt force power, but it does seem to have an effect. We also think that these antibody responses predate the treatment interruption at sort of a very weak level, and then they're boosted with antigen returning, with virus rebounding, and they have differential responses against some viruses versus other. And so some of the questions that we think are interesting that we hope to continue to work on is, which of these reservoir or rebound variants are being targeted? Are these early variants that have retained the earliest antibody responses from these individuals? Are these more recently circulating variants, which have sort of more recent B cells that are primed against those specific viruses? Or is there something unique about perhaps the viruses that have the most presence during suppressive antiretrovirals? Maybe there's an expanded clone that's releasing a lot of one particular type of virus, sort of priming a specific antibody response over antiretrovirals. There are a lot of biologically possible ways to think about how these antibody responses could persist through suppressive ART and be relevant at the time of treatment interruption. I think another important question is, what is the clinical significance of these very modest neutralizing antibody responses? Can they be harnessed in any way to work in synergy with exogenous antibodies? Can they be boosted through vaccination or through other immunomodulation to make them have a more important role in restricting virus replication and therefore perhaps working in concert with other approaches? And I know that we are interested in this, and there are others in the field also working on these exact topics. So the next immune response that I want to talk to you at the time of rebound is slightly more robust and a little easier to feel confident about. And that is the role of type 1 interferons at rebound. And so this is a collaboration with Beatrice Hahn, who is just down the hall from me at the University of Pennsylvania. And our interests have aligned recently with my interest in rebound and her interest in the sort of phenotype of viruses throughout the life course. And so Beatrice's previous work has shown that transmitted founder viruses are uniquely resistant to type 1 interferons. And she's shown this in several different cohorts, but I think one of the most interesting is looking at donor-recipient pairs. So these are acutely infected individuals in whom the donor or their partner who was infected with HIV, in this case with a chronic HIV infection, had their virus sampled near the time of transmission. And what we can see from the chronic viruses, so these are virus isolates or infectious molecular clones derived from, let's see, 7 distinct chronically infected individuals. And then these viruses, these isolates are tested for their interferon sensitivity. So they are tested in dilutions with different concentrations of interferons, and it's determined the concentration of interferon that inhibits 50% of replication, and that is considered the IC50 for in this case, interferon beta, and then that is plotted here. And so what we're looking at in each one of these little pink dots are multiple chronic viruses sampled at the time of transmission to an acutely infected recipient. And what you can see is that the chronic viruses are relatively sensitive to interferon beta, whereas all of the acute viruses are substantially more resistant to type 1 interferon. And this sort of reflects the process, the biological barrier that these transmitted founder viruses have to go through as they cross the mucosal barrier, as they establish infection in a new host. They have to overcome an innate immune response, and therefore being resistant to these type 1 interferons is very important for their success in this context. So this was published several years ago, almost 8 or 9 years ago by several different groups. And this is the sort of foundation for the next step. So what we did recently was we looked at a cohort of 10 longitudinally sampled individuals. This is from a cohort in England in whom 10 individuals had their viral loads and CD4 counts captured over weeks of infection until they were put on antiretrovirals, which is shown here in gray. And we have 2 or 3 different phenotypes here. So we have sort of our traditional progressors. We have 2 individuals who had very slow progression, and then we had 2 rapid progressors. And if we take the viruses sampled from acute infection through chronic infection, we can look at the interferon resistance over the natural history of HIV infection and get a sense for the kinetics of interferon resistance. So in our 6 sort of traditional or normal progressors, what we see is what we call the SMILE phenotype. So all of our transmitted founder viruses are quite resistant to type 1 interferons. And here we're looking at interferon alpha 2, and on the right here, we're looking at interferon beta, but you can see our viruses start out quite resistant to interferons. They drop over the first 6 months to a year in their interferon resistance, and while there's this clinical latent period of early infection, they maintain a low level of interferon resistance, and then that resistance rises a little with clinical progression, with CD4 decline, until this sort of historical cohort met indications for ART initiation, and they were started on antiretrovirals. This group right here of 2 individuals who were very slow progressors. So again, we start with more resistant virus. It falls pretty rapidly, and it doesn't rise again as there's no clinical progression in these individuals. And finally, we have 2 rapid progressors in whom we see high levels of interferon resistance, and that never really falls over this first period of, over this sort of highly inflamed, highly cytokine-driven rapid progression where we maintain very high levels of interferon resistance. So the kinetics of the interferon resistance is fascinating in my mind because it really shows how plasma virus is this snapshot of what pressures are being placed at that moment on the virus. So it's a balance of all of the immune pressures, whatever drug pressures, and in balance with the intrinsic viral fitness of that virus. We see universally across these 10 individuals that transmitted founder viruses are highly resistant, and that again reflects this intense innate pressure at transmission prior to adaptive immunity. We see that interferon wanes early over the first 6 months of infection, and in the studies that characterize this, you can see the virus sort of trades off adaptations to deal with, it selects for escape mutations to antibodies and to T cells as the interferon pressure becomes less. And then finally, we see interferon resistance rising again with disease progression and CD4 decline. So in some of these individuals, we had the opportunity to look at viruses sampled during ART suppression. So here is an individual MM40. They had several years of natural infection. We see the nice smile. They're put on antiretrovirals. More than a year later, we have a virus outgrowth sample from the reservoir. So again, this is like a replication competent virus, much like the QVOA viruses we talked about a little bit ago. And we can see that the phenotype of this virus is very similar. It's sort of in the mid-range here, but it's a relatively sensitive virus, and it actually phenotypically matches with the viruses that are sampled right before ART initiation. And if this is a phylogenetic tree of this virus evolution over time, here's our acute viruses in blue, and then over time we see virus diversification. And what you can see in purple here, this star, this is the sequence, the envelope sequence of the reservoir virus, and it matches both in sequence and in phenotype with these viruses sampled near the end of the natural history, right before antiretrovirals are started. And we see 4 other individuals in whom we were able to sample from the reservoir, and we see the same phenomenon. The phenotype matches with the phenotype of viruses just before treatment interruption, and then they're modestly interferon sensitive. And that is the case for all of these individuals. So the next thing we wanted to ask was, what about rebound viruses? So again, we were able to look at this trial, this A5340 study. And again, here's A09, this participant that I forced you guys to stare at this phylogenetic tree before, and we're staring at it again. But we were able to take these multiple rebound virus populations and compare them with our multiple outgrowth viruses and get a sense for what the difference was in interferon sensitivity. And remarkably, our rebound viruses were substantially more resistant to interferons than any of our reservoir viruses sampled either before or after in this individual. And in fact, these rebound viruses were really strikingly resistant to type 1 interferons, more than we've seen in other clinical contexts. And so when we look through multiple trials that we conducted here, so we have no intervention trials, we have trials with VRCO1 and 3BNC that we talked about before, as well as trials where we administered exogenous interferons. What we see is that rebound viruses in pink compared to reservoir viruses in blue are way, way more interferon resistant across the board. Universally, it is a defining feature of the rebound viruses that we have sampled. We have not yet sampled a rebound virus that is not quite resistant to interferons. And if we put all of these populations together and we look at our rebound viruses in pink, our reservoir viruses in blue, our historical cohorts with our transmitted founder viruses in this nice sort of teal color, our early chronic viruses here, our late chronic viruses here, you can really see that our pink rebound viruses are uniquely interferon resistant compared to our others. That's compared certainly to our reservoir viruses, but also to transmitted founder viruses, which previously had been the most sort of interferon resistant viruses that we had sampled. And so this suggests that there is a really strong innate selection pressure in the tissues from which virus is reactivating during treatment interruption. And it may be even stronger or different than what we see in the transmission process, where we have a well-mapped and characterized cytokine storm through which these transmitted founder viruses have to break through. Okay, so very briefly, that was published in a paper we just got published a couple months ago, but what are we working on now? So I'll say one of the things we're revisiting is this concept of reseeding. And if you look at our published paper, we actually show that there's 2 post-ATI reservoir viruses from A08, one of the subjects from the study we were talking about, in which we actually have elevated interferon resistance from the reservoir. And this is an example here of reseeding. So we do believe that these viruses are very closely related to the rebound viruses, and they became a part of the reservoir and then were sampled later on. So this is A08 here, and this is another participant, A13, and after doing more sequencing and more sampling of the reservoir viruses, we found 2 examples of reseeding in each of these individuals. So here's A08, where we have both red rebound viruses and blue virus outgrowth viruses co-mingling in this lineage, very closely related. Now this is just envelope. If we look at the whole genome, there are slight differences in the genome across non-OMV genes, but with an envelope, they're basically identical. And we see the same thing down here. In A13, we also have identical sequences in envelope of 2 distinct lineages. If we look at the whole genome sequence, there are a few minor changes within those viruses. So they're not completely identical, but they're very similar to what was sampled very early on. Now, if we dig into this A08, for example, and we look not only at the sequences, which are identical in envelope, but not throughout the whole genome, if we compare the IC50s to interferons, our virus outgrowth viruses, they have a heightened or higher levels of interferon resistance, but it doesn't quite reach the level of our rebound viruses. So against alpha-2, it's very similar. Against beta, it's not quite there. But we can also characterize other phenotypes. So we can look at the cellular tropism, and in this particular individual, there's a very diverse sort of heterogeneous reservoir. And in fact, within our rebound viruses, we see examples of both CCR5, CXCR4, and dual tropic viruses. So there isn't sort of a specific cellular tropism that's a determinant of being a rebound virus. And we can also ask whether these viruses are able to replicate in monocyte-derived macrophages. And this is a particularly interesting component, I think, of understanding these rebound viruses, because there's a lot of discussion of whether rebound viruses maybe arise from a macrophage lineage cell. And if that's true, it would seem that they should be able to be replication competent within macrophage lineage cells. That doesn't determine or doesn't, say that they're automatically from a macrophage, but you would think that that would be associated with that phenomenon. And what we see is that rebound viruses in this one individual, some of these rebound viruses are in fact able to replicate in macrophages, but others are not. And so when this is just one particular experiment, we've of course tested in many different donors, but when we look at these viruses, panels of rebound, as well as virus outgrowth viruses, as well as sort of represented macrophage tropic sort of lab strains, we see that we do have many rebound viruses that can replicate in macrophages of different donors. And if we compare across these groups, we see there's sort of an enrichment for this property, but it's not universal. So not all rebound viruses are able to replicate in monocyte-derived macrophages, and we're sort of digging into this now to figure out exactly what the determinants of macrophage tropism are, because it is not just a one-to-one ratio with interferon resistance or being a rebound virus. And so this is something that we're actively working on. Interestingly, when we take these rebound viruses that are incredibly interferon resistant when they're tested in primary CD4 positive T cells, when we look at them replicating in monocyte-derived macrophages, and we give various concentrations of interferon alpha-2 or interferon beta, we do not see a distinction in their sensitivity to interferons compared with chronic or acute viruses. So this interferon resistance seems to be a property of replication in T cells and not across other cell types. And so we're also digging into that. Another thing we're very interested in is, as we wanted to map sort of the autologous antibody response over time, we also want to know what happens to these interferon pressures or the interferon resistance of these viruses over time. So in that historical ATI study where we had long periods of replication after treatment interruption, we were able to track these rebound viruses. And what we found is over time, after weeks and months of replication, they lose this property of interferon resistance. So much like what we see in acute infection going into early chronic infection, this interferon resistance is really important for the early recrudescence and replication at the first treatment interruption, but these viruses lose that interferon resistance over time. And so we need to look at this we need to map this in sort of more fine detail and in more participants, but this is something we're also interested in. And finally, the presence of some of these reseeded viruses or these matched reservoir and rebound viruses that are genetically very similar enable us to be able to do mapping of the determinants of this interferon resistance. So here's just two examples of sort of matched viruses in which we have rebound and outgrowth viruses, which are genetically similar. They differ by between 2 and 5 amino acids throughout their genomes, and they have quite distinct interferon sensitivities. And this allows us to sort of map the viral determinants of interferon resistance. And we also have some unique systems so that we can use these paired viruses in an unbiased CRISPR screen to sort of assess what interferon stimulated genes could be driving this interferon resistance as well. So in summary, I hope that I've been able to convince you that rebound viruses are an important subject for study, as they are a direct link to the clinically relevant reservoir, and therefore it's important to understand both the viral and host determinants of that phenomenon. The disconnect between the sampled reservoir and rebound viruses challenges current cure trials, and it suggests mechanistically that rebound viruses are either rare and compartmentalized within the reservoir that is not currently well sampled, and or they undergo intense selective pressure at ATI that forces them to be either selected rare variants or rapidly adapting to those immune responses during treatment interruption. We think that the interferon resistance of rebound virus is a really salient feature of these viruses, and we're working right now to sort of better define that biology. And so we're working to comprehensively characterize the full phenotype of these rebound viruses, the frequency of reseeding in these treatment interruption trials, and as we entertain different types of ART restart and clinical trial structures, see how that changes, to look at the kinetics of these interferon resistance over time, and finally, to use these paired viruses, which have very similar genetics but distinct phenotypes, to be able to map both the host and viral determinants of interferon resistance. So there's lots and lots of people to thank for all of this work. This last part was a collaboration with Beatrice Hahn and her group. And of course, the study participants that participate in these studies make all of this work possible. And so I need to be very thankful of them. All right, like 30 seconds left for questions.
Thank you very much, Katie, for quite a tour de force of, I guess, viral resistance to host immune responses. Are probably worryingly good at it. Thank you also for staying up so late to present your work. I think it's nearly midnight there now, so if you'll indulge us, maybe there might be a couple of questions. I guess you kind of touched on my first question in your conclusion slide about to what extent the resistance that we see is coming from the reservoir or is adapting very, very rapidly. I mean, both the interferon resistance and the fact that you tend to see unusual clones could be that there's just extremely rapid selection. Is there a way to address that, do you think, in these studies?
Well, yes, I think that there are multiple ways that we can go at it. Some of the things that we're trying to do, and I don't think that the explanations are mutually exclusive. I don't think that it's just that they're rare variants that are selected for, or that there's rapid adaptation. I think that the evidence suggests that probably there's some combination of both of those things happening, and that virus, I mean, if I had to project, I would say viruses probably need to have some level of interferon resistance, and they can accrue some finite amount more through rapid adaptation in those first days to weeks of virus of replication in the tissues before they're sampled systemically. So some of the things that we can do to sort of assess for that is to be more thoughtful about how we select from the reservoir. So we can look at we can look at distinct cells, we can look at distinct compartments. We can take the same cells we've been sampling and, for instance, do virus outgrowth assays in the presence or absence of different interferons and other cytokines, thinking that if we put these cells in the sort of conditions that are most like what is happening in vivo reactivation, can we enable viruses that wouldn't otherwise reactivate from those cells? And we do see some preliminary evidence in virus outgrowth assays in the presence of interferons that we actually don't increase the number of viruses that we retrieve, but the phenotype is different. So we see sort of this intermediate grade of in between sort of modestly interferon resistant virus. And so, so we're doing more of those sort of interferon treatment of virus outgrowth to see if we can sort of recapture sort of the in vivo phenotype in our ex vivo assays a little bit more.
I guess that rapid adaptation also has implications for interpreting the macrophage tropism, because it could be that a virus rebounded in a macrophage and then, rapidly switched to T cell tropism because that was what the major substrate for growth rather than where the virus actually came from.
Yeah, I think all things are possible. It does seem unlikely. Well, I guess it's possible. If we're saying these viruses are rapidly adapting in one way, there's no reason that they couldn't also sort of adapt in another way. But it seems more likely to me that they would retain some type of macrophage tropism if they're just a few replication cycles away from truly being an efficient replicator in that system.
And I guess the big elephant in the room in HIV antibody studies that no one seems to be paying much attention to is the AMP trial or trials in South Africa, which showed no efficacy of VRC01 given over months to people. Do you have any thoughts on that? In transmission, I should say, where we assume that there's a very narrow bottleneck of virus.
Right. So, I mean, I think that you can interpret the AMP studies as glass half full or glass half empty, depending on how you want to look at their sort of sieve analysis, where they said there was a deficiency of really sensitive viruses being transmitted. Therefore, they, inferred effect against, or, efficacy against sensitive viruses. I mean, I guess my experience with this A5340 study is the vast majority of individuals with chronic infection were not able to be suppressed even a tiny bit with this VRC01 antibody. And I think that the levels of antibody of VRC01 resistance are in transmitted viruses are also quite high. So I don't know that the AMP study is super surprising in that regard. And I think the clinical trials field has moved on to antibodies that are 10 to 100-fold more potent than VRC01 by themselves, and really thinking about using them in combinations. So I think the positive spin from the AMP study would be that we saw perhaps some efficacy against very sensitive viruses, demonstrating maybe proof of concept. And then you take that proof of concept and you say, if we use combinations of much more potent antibodies, that would translate to a much more broad effect. I think that's the positive view of how to, you know, move forward from the AMP study that didn't show broad protection against transmission.
Sorry, I've just caught up with the online questions. I better ask someone else's. Tony Kelleher asks, does the behavior of the virus allow you to speculate on where the rebound reservoir is?
Yeah, so I mean, I think that's kind of the goal of looking at macrophage tropism, and we were initially kind of skeptical about the role of macrophages as potential sites of replication, but there does appear to be enhancement for monocyte-derived macrophage replication, and so that seems to be one of the conditions that could be met. I think that the most commonly implicated monocyte lineage cells, however, for a persistent reservoir are these tissue macrophages, which are quite distinct from our monocyte-derived macrophages that we study in the lab. So we're trying to work with groups that have a much better handle on tissue macrophages. There's several groups at Penn that have in like ex vivo as well as sort of induced pluripotent stem cell models of tissue macrophages for us to get a better sense of how these rebound viruses, how they sort of replicate, how they function in those more really macrophage reservoir-like cells. I do think compartments is always an issue. The central nervous system is probably the most likely source of a persistent macrophage reservoir. I mean, tissue macrophages are everywhere, but there's really good evidence that they're important in the CNS system as well. So I think that working within animal models, particularly the non-human primate model, to get an understanding of those brain macrophages is going to be also important. And I think there's a lot more to go in that regard as well.
Yes, once you say tissue macrophages, you kind of like open it up to anywhere. It becomes even broader than if it were in T cells, I guess.
Yeah, I mean, there's quite a bit of variety of tissue macrophages, and they obviously have implications in lots of cells from the lungs, or lots of tissues from the lungs to the genital tract to throughout the body.
Well, it's past the hour, so we probably should leave it there. And I'll just thank you very much again for sharing your very exciting work on the reservoir and for staying up so late. And I guess if people have any other questions, they might send them by email. So please join me in thanking Professor Bar very much again for joining us today. Thank you.
Thank you.